Hello, grade 10. Here we are with lesson number 8, translation. So today we're going to be doing the second part of our protein synthesis, where we went last class through the overall process, a central dogma of molecular biology, and then we talked about transcription. Today we're going into translation. So our targets for today are I can explain the process of translation, and I can use a codon chart to identify the amino acid coded for by the mRNA. We are going to start today, though, with some review of transcription, just to make sure we got it well for yesterday, last class. So here are a bunch of terms on the left and our words, and we have statements that go with them on the right. So take a minute and match them up. So for example, I know 5' to 3' is the direction in which mRNA polymerase works. So you can match them. So pause the video and finish out the matching yourself, please. Okay, so I'm assuming you've done all the matching activities and you can now check your work. Here are the answers. So I rearranged them to show which ones go with which. So again, pause the video, check your work. Okay, now that we reviewed transcription, it's time to dig into translation. So translation is all about using these codons. We're going to start with talking about codons to give some context here. So codons are 3 base long sequences. We can see here AUG, GGA, GUU, these are the codon, okay? They're 3 bases long, and each gives a specific amino acid. So there's 20 amino acids. Here are some examples: methionine, glycine, valine, and they're the building blocks of the protein. So the codons code for the amino acids, okay? And that's happening in translation. So GGA, which we see here, that one codes for glycine. GUU here codes for valine, that kind of thing. AUG here, this is a special one. It's called the start codon, and it tells the ribosome to start translating. It also codes for methionine. So if it occurs very beginning of the messenger RNA, then it's going to be telling the ribosome to start. And if it occurs later in the RNA, it just means methionine, just that particular amino acid. There are also 3 stop codons, and they tell the ribosome to stop doing translation, to stop matching up amino acids with mRNA codons, and they are UAA, UAG, and UGA. There are 3 stop codons, and they say, okay, we're done. The entire gene has been translated, time to stop. So in your foldable here, you have some boxes for translation. You can take your notes there if you like for this part, or you can choose to get over your notebook or your laptop and then come back and fill in the diagram later. But you should have a complete foldable for next class. Okay, so next week. After transcription, so we have our mRNA here has been produced in the nucleus, going to leave the nucleus and go in the cytoplasm. And our ribosome, here we see it's colored in green, is going to bind to it. Remember, ribosomes are made of rRNA. And here you all can also find the tRNAs. They're here, the little pink ones in this picture, the transfer RNAs. And they're all attached to a certain amino acid. We can see here this one has met, valine, glycine, different ones attached. Here as well, amino acids attached to tRNAs. What transfer RNAs do is they transfer amino acids based on the sequence on them. So what happens when the ribosome, when it finds a start codon, here we have AUG, the start codon, that's going to tell the ribosome to start translating. And it's happening, of course, at the 5' end. Everything starts from 5 to 3 and begins to translate to form what we call a polypeptide. Now, polypeptide is just a chain of amino acids. You see here's the label polypeptide in your foldable. Okay, just amino acids all in a chain bonded to each other here. This is happening when the codon of the mRNA here, when it matches up with the anticodon here in the pink of the tRNA, and that tells it to release that amino acid. So because CCA matches up with GGU, that tells the tRNA to release the amino acid proline or pro. And then before here, we had glutamine from the codon CUC, and the anticodon was GAG. So by matching it up using complementary base pairing, again, it's going to make sure you release the correct amino acid. Now, how do we know which amino acid is which? We're going to use a codon chart. So we have sets of these in class, and if you need to use it on a test, you'll be given one either in the question or you can use the actual codon chart. Those we have in class have this rectangular format on one side and the circle on one side. You should know how to use both, but everybody has a preference. I really like the rectangular one, some people like the circular one. They both do the same thing. So how these work is it tells you, in here is the codon, which is in the messenger RNA, and I just find the codon sequence, and it tells me what amino acid is. So here we have amino acids as a 3-letter code, so THR-ALA. They have full names, like ALA is alanine. You don't need to know that, okay? It's okay just to call it ALA in the 10th grade. They also have a 1-letter short form, just A, just T, just P, just S. And those are for when we have really, really long polypeptides and we don't want to write 3 letters. So we just write 1 letter each to keep it a little bit more manageable to work with. You can also see here we have the start codon in green and the stop codon here. Stop codons in red. To read this one, you want to look here in the column. This is the first letter is going to be, first letter is the row. So these are all begin with U. These all begin with C. These all begin with A. And these all begin with G. And then the second letter is U, C, A, and then G. And then you just find the third one after that. Okay, so U, here we go, is PHE. Okay, UCA, SER, that kind of thing. On this one, you start with the first letter in the center. So if it begins with a U, you start here. And then the next letter, let's say it's a C, you start here. And the last letter is an A, it's here, UCA, and that's serine. So you notice how a lot of times we had the same amino acid coded for by multiple codons. So here we can see there's 4 that code for serine, and there's 4 that code for serine here as well. You can use either format, the circle, first letter, second letter, third letter, if you want to, or you can choose to use the rectangle. Either one is fine. So let's practice. Here is, I'm leaving the circle codon chart up for you guys, and here are some codons I want you to try with. So just look at the codon chart, look at the codons here, pause the video and complete it. Okay, so I'm assuming that you have completed your practice. Here are the answers. So GGU is glycine, GGU, gly. CAA, GLN here. UAA is a stop. AAA is lysine. AUG, methionine, or the start. And GUG is valine, or GUC is valine. Okay, so you can see just reading the codon chart what something you need to practice. Again, we will always give you a codon chart. You don't need to memorize it, but you do need to know how to use it. Okay, so what is this codon chart talking about? These letters, ALA, PRO, these are all the amino acids. This is a table that shows the structure of all 20 amino acids. You don't need to know the structures of all of these in the 10th grade, but I want to just show you that they have a similar kind of base structure, and the part that's highlighted is what's different. So they all have different chemical structures, and that's kind of what helps make the protein have its shape and have its function, is based on which amino acids come together to make the protein. So we can see here we have the messenger RNA. We have the tRNAs with their anticodons on the bottom, matching up with the codons on the mRNA, and each tRNA is releasing one amino acid. It's making this long amino acid chain we call a polypeptide chain. And we can see here, kind of zooming in, this is just pro, lu, gly, il, asin, cis, just in order, those amino acids. And the order of them does matter very much for how the protein will have a specific function. And that's why we need to have the DNA message transcribed to messenger RNA to be translated into a polypeptide chain for the protein to have a proper function, because the order of these really makes a big difference to protein shape. And you'll learn why that is the case with further study of biology. So let's practice some more. So you're going to take the sheet you've been given, and you're going to take the DNA here. I've given you just the template strand, and you're going to first figure out what the messenger RNA is using complementary base pairing. Once you have that and you have all the codons, you're going to figure out what the anticodons are, and then using your codon chart, you're going to figure out what the amino acids are. Now, if you don't have a codon chart with you because you're not in class, just Google codon chart. There are a million of them, or you can take a screenshot of the previous slide. Okay, I'm going to post the slides as well for you guys. So let's practice together. So if I have a base T in my DNA, my mRNA is going to be an A. If I have a C, it'll be a G. A C, it's a G, G, C, C, G, G, C, C, G. A goes with U because it's RNA, not DNA. G goes with C. A goes with U again. G goes with C. C goes with G. T goes with A. A goes with U, and G goes with C. So those are my codons. That's the mRNA, right? So these are all codons. Then to figure out the anticodons in the tRNA, we're going to use complementary base pairing between the mRNA and the tRNA. So A goes with U, G goes with C, G goes with C, C goes with G, etc., all the way across. Now, to figure out the amino acids, you have to use the codons. So we're going to be using these letters for the amino acids, not the tRNA, okay, because we're matching to the codons, and the tRNA is just how it matches, okay? So use your codon chart. That's why it's called a codon chart. We're using mRNA codon. So AGG gives us arginine. CGC gives us arginine again. GUC gives us valine. UCG gives us serine, and AUC gives us alanine. So take a minute, pause the video, and now you do the second one by yourself. You have these on the sheet that was handed out to you. Okay, once you're done, start the video again, and here are the correct answers, so you can check your work. So pause the video, check your work. Okay, we're going to move on. So next part, translation. As we're adding each of these amino acids in the chain, the ribosome is going to make what's called a peptide bond. So a peptide bond is a type of bond that's connecting the amino acids here to make the chain, which is part of why it's called a polypeptide, many peptides together. Once that's been made, the tRNA that had that amino acid, like this one here, is now empty. It's going to leave the ribosome and go get, like here's an empty one as well, and then go get the right amino acid put onto it again. And that's going to be done in a process you will not learn about in the 10th grade. When there eventually gets to a stop codon at the very end, it will stop. It will terminate translation, and the ribosome will be completely disassembled, taken apart. There's a big part here of the ribosome and a small part with a large subunit and a small subunit. They'll kind of come apart and release in the cytoplasm. And this polypeptide is going to be released to get processed and folded in the cytoplasm. That folding usually means making really small change to the protein shape. And we're not going to talk about how that happens in the 10th grade, but there are a few different things that can be done, usually small tweaks to the structure, just to make it have the perfect shape, because the shape of a protein is very important for a protein to be able to do its job. And now you've gone from DNA to messenger RNA to protein. Boom. So you're going to log in and practice this. So here, protein synthesis, you're going to use headphones for this, and the link is on Classroom. And you're going to be able to click and actually do like the matching part to transcribe antigens at a gene. It's meant to help you review, and you can definitely use screenshots if you were taking digital notes to help supplement your notes, make observations, maybe use it to help add to your foldable. Just use it to help supplement your notes and practice more and more. It's going to take a few different practice activities to really get this process clear in your mind and to memorize it and understand it really well. Once you've done that, we also have a sort activity you can do with a partner, and try to do it without your notes. You have a bunch of cards and you're going to put them in the correct order, and then if you finish the correct order, let me know and the teacher can check it for you, and then you will be able to see if you have it correctly, and if you do, take a picture of it, put it in your notes. Okay, lots of chances to practice this because it's really going to take a lot of practice to get there. All right, that's the end of our lesson. Hope this was helpful for you guys today, and yeah, take care. Happy studying.